DNA or deoxyribonucleic acid is nucleic acid polymer produced from covalent bonding of nucleotide monomers that contain the sugar deoxyribose. That is the genetic material of nearly all organisms. Additionally, more than hundreds and thousands of DNA is required to make organisms function. That is why DNA goes through a process called DNA replication, in which a parental DNA replicates itself into two new identical DNA to the parent strand. The DNA structure includes a nitrogenous base and paired links of adenine and thymine or guanine and cytosine. DNA replication requires three main steps: unwinding, complementary base pairing, and the joining of the new strand. In unwinding, DNA helicase enzyme unwinds DNA and separates the parental strands. This creates two replication forks that separate. Each fork is actually joined to another form of a replication bubble. The separated strands are now used as a template to create two new DNA molecules. Single-stranded binding proteins attach newly separated DNA and prevent it from reforming the helix so replication can proceed. Complementary base pairing, DNA primase places short RNA primers on the strands to be replicated. DNA polymerase recognizes this RNA and begins DNA synthesis, allowing new nucleotides to form complementary base pairs with old strands to form a chain. Parental strands are anti-parallel to each other. That means one strand goes from a 5 feet to a 3 feet and the other 3 feet to 5 feet. The new daughter strands must also be anti-parallel to its matching parental strand. The 5 feet to 3 feet strand is called the leading strand and is exposed so that synthesis is easier and replication is continuous. The other strand in the fork must be synthesized in opposite direction, requiring DNA polymerase to synthesize the new strand in short 5 feet to 3 feet segments with periodic starts and stops. The strand is called the lagging strand. Replication of this strand is made in segments called Okazaki fragments. Joining begins after the strands are made. DNA polymerase converts short RNA sequences into DNA. DNA ligase is like glue in which it mends all the Okazaki fragments together, resulting in two double helix molecules that are identical to each other and its original molecule. Prokaryote DNA replication goes through a process called the origin of replication. Another note to add is that telomeres are the caps at the end of each strand of DNA that produce chromosomes. Telomerase is a ribonucleoprotein that adds polynucleotide TTAGG to 3 feet end of telomeres. In the event that DNA mutates, whether by deletion, insertion, or substitution of particular sections, nucleotide excision repair removes DNA damage induced by ultraviolet light, which may be by such mutations.
Thank you.